Okay, good morning, good morning, good morning. This is Screencast 12 of section 6.6, and we have two little things to cover here. One is the role of the sex hormones in puberty, and the other is in vitro fertilization. So, with the hormone testosterone, we'll go to the male first. We've already established that it is responsible for the development of testes, prenatal development of testes, and it is directly involved in sperm production following the onset of puberty. It also aids in the development of secondary sexual characteristics, including the following. Let's put them all up there for the male. All right. The growth of facial hair, and enlargement of the larynx, coming with a deeper voice, increased muscle mass, enlargement of the penis, helping to maintain male sex drive. That's also should be considered there. A female, again, for a female, estrogen and progesterone, again, cause prenatal development of female reproductive organs and drive her secondary sexual characteristics. And of course, all right, including the growth of hair and underarm hair. So it's different for male and female. You see other secondary characteristic, enlargements of the brain, widening of the hips, and that is to accompany a baby, perhaps. Oops, sorry, let's go back here for just a second. So again, you want to know the primary. Again, the development of sex organs prenatal, sex cells, and of course, secondary sexual characteristics after the onset of puberty. All right, lastly for this, we have a review because we kind of covered this in genetics, but we'll go a little bit more depth now. Okay, in vitro fertilization, again, is a genetic technology technique that occurs outside the body. So in vitro actually, I think, literally means glass or in glass. So how does this work? All right. So, and why does this work? Some of the reasons why. Again, approximately 1 out of 6 couples are infertile. All right. And in vitro fertilization is generally used as a way to overcome female infertility, physiological infertility for her. All right. Including blocked oviducts, fallopian tubes, or timely ovulation. As far as guys is concerned, there is infertility affecting sperm count, but normally you can isolate the number of sperm that you need and get that done in vitro wise. All right, also can be used. All right, so in vitro can be used for sort of low sperm count, as we just said. So what actually happens here? We're going to give a little more information and go over. So it involves, of course, removing an egg, in vitro fertilization in a petri dish, glass petri dish, giving it the conditions to start to divide and then put it back into the uterus. All right, there's a little bit more to that. So we're going to go over it a little bit more now, the in vitro process. There's about 8 steps. So you've got to first start regulating the menstrual cycle. So drugs are given to stop the menstrual cycle. Again, that would be the regular secretion of FSH and LH. You do not want to have an egg produced. All right, and so by stopping the cycle, you can take control. Doctors can take control. All right, of that. So then large doses of synthetic FSH are given to develop more than one follicle, about a couple of weeks worth of that. And this is usually done, fun fact, in the form of a spray into the body there. All right. And then a hormone called HCG, human chorionic gonadotropin, is given about 48 hours before the collection of eggs, forcing the follicles to have the eggs mature. That's getting ready to ovulate. All right. Somewhere in here, again, this can be provided anywhere. Somewhere in here, the male had to make a deposit. So I just put it at number 4, but it could be further down as well. But that deposit is necessary. Okay. Eggs then would be extracted via aspiration needle through the vagina prior to rupturing. Okay. They would be fertilized. Okay. And incubated in the presence of a sperm sample of a male donor, and then analyzed. I suppose there has to be some analyzation to see if fertilization is taking place. The embryos that you wish are selected, placed in the uterus, and wait approximately 2 to 3 weeks to see if implantation has successfully taken place before the woman will start to take further hormone. All right, so scan for any developmental issues as well. So the procedure again, stop the normal cycle, hormone treatments to promote ovulation, super ovulation because you need a lot of eggs, extract multiple eggs from the ovaries, sperm collected somewhere there. Fertilization occurs externally, that's the in vitro portion, implantation into the uterus, and then test for pregnancy after a couple of weeks. All right. So in this case here, we then finish with discussing the pros and the cons. This goes back to the pros and the cons of genetic implantations. So just throw these up here for a second. Again, you see some of the pros. Environmental infertility, something going on outside that will not affect the offspring. There's no pain to the embryo. There's no nerves to the embryo yet. You can genetically screen before transferring, and this will help parents who have a strong desire for their child and don't want to adopt. Against, you said you have to collect extra embryos. What happens to those? Normally, those are frozen. That's expensive. This often results in a multiple pregnancy, sometimes 2, 3, 4, and there have been instances of more because you have to put multiple eggs in because many of them don't implant and you're looking for one. So going on there with a high rate of failure. So it's expensive. Often I think for one in vitro it can be upwards of $30,000 to $40,000. So there are some ethical issues before and ethical again. So when we deal with ethical issues, you're to choose one that you can speak to, strong desire for a child, expensive and high rate of failure. And again, high rate of failure goes back to why you take out multiple eggs and have fertilized multiple eggs are implanted. All right, so this then ends 6.6.